And okay, let's just hope that the internet will cooperate because this is quite long. This is around 56 slides. So we are now in the brain. Oh. So this is how the brain of a dog looks like. So it has the cerebrum, the cerebellum, then we have the glands here, the hypothalamus, the thalamus, and even we have the pituitary over here. So you have your, and the pineal gland, which we have discussed on breathing. And then we also have the brain stem over here and the beginning of the spinal cord. This is just how it looks like. So the brain, and this brain again. And these are basically the parts of the brain, which you will later have to be familiar with. So we are now in the central nervous system. So the CNS is formed from the ectoderm, germ layer of the embryo. So you've already known that in breathing. And the anterior end of the neural tube becomes the brain, while the remaining parts will develop into the spinal cord. So we get to know this even more in embryology and the development of this. So this is how it looks like. So you will get to know this even further when you go to embryology. I'm just giving you a preview. So function of the brain, so it controls and coordinates all the activities of the normal body. And the brain is encased by the cranium that serves as its protection from mechanical damages. And it is divided into three regions: the forebrain, the midbrain, and the hindbrain. So the cerebral cortex is part of the forebrain. So the cerebrum, or the telencephalon, so this takes up most of the forebrain and contains 90% of all neurons in the entire nervous system. And it is divided into two hemispheres, the left and the right. The two hemispheres are linked by the corpus callosum, which is a tract of white matter found in the roof of the third ventricle. And this hemisphere's surface are deeply folded, which enables a large surface area to be enclosed within a small cranial cavity and allow nutrients in the cerebrospinal fluid to reach the cell bodies of neurons lying deep inside the tissue. So parts on the surface of the cerebrum, you have the gyri, also known as convolutions. So these are the numerous folds on the surface of the cerebral hemispheres, greatly increasing the cerebral cortex surface area. And we also have the sulci, the shallow grooves between the gyri. So these are your gyri. So these are the hemispheres. So it's divided into left and the right. The more gyri the animal has, the more well-developed is its brain. We'll go to the human as an example. Humans with dementia and Alzheimer's disease, like my grandmother, their MRI, magnetic resonance image of their brain, the sulci and the gyri are less pronounced. So meaning they're losing some parts of the, it's a nerve degenerating disease, and it's usually affecting the brain. We also have fissures, which are deep grooves. So you can see fissures also here. So there's fissures. But this one is a long fissure, longitudinal fissure, which divides the cerebrum into two hemispheres. So the lobes of the brain are named for the bones they underlie. So they have four. So we have the frontal lobe, occipital lobe, temporal lobe, and the parietal lobes. The cerebral cortex have been mapped for sensory and motor areas. And the right hemisphere controls the skeletal muscle movements on the body's left side, while the left hemisphere controls the right side. So if you are an orthodox or a right-handed person, it's actually your left side that's controlling it. And if you are a southpaw or left-handed, your right brain usually is the one that controls it. And they also have specific points on the motor cerebral cortex that controls specific muscles. So these are the lobes. So you have the frontal lobe, the parietal lobe, occipital lobe, and the temporal lobe. So these are the ventricles, by the way, and this is the corpus callosum, which I was talking about. So this is the third ventricle is this one, which you will later find out in the coming slides, and this is the corpus callosum over here. So the frontal lobe, rostral part of each cerebral hemispheres, and the caudal part of the frontal lobe houses the motor area of the brain. Also, in the frontal lobe, you will have the occipital bulbs. Olfactory bulbs. Sorry, olfactory bulbs. So that is where you can find. That's where the sensor for the sense of smell. It's in the frontal lobe. So particularly the olfactory bulb. The parietal lobe is the dorsal part of the cerebrum, and the rostral part of this one is the area for temperature, pressure, touch, and pain sensations. Ventral cerebrum, that's where the taste and smell has. So you can find there also the olfactory bulb. Association areas, these are areas next to the sensory and motor areas, and they analyze a sensory input and deal with memory. Lateral ventricles. The two cerebrospinal fluid-filled spaces of the ventricular system, and each surrounded by a cerebral hemisphere. And we have the third ventricle, which encloses the diencephalon, which encircles the interthalamic adhesion. So you already saw the third ventricle. So here you go. This is the third ventricle over here. And right below it are some of the glands, like the thalamus, hypothalamus, and the anterior pituitary, or the pituitary glands. So, sorry if I'm yawning. Okay, cerebral cortex. So this is the outer layer of the gray matter. It contains millions of cell bodies of the neurons. It has a basal ganglia, a group of cells found deep in the brain. And the basal ganglia includes the caudate nucleus, the putamen, and the globus pallidus. These ganglia act as relay stations collecting information and sending out impulses to a variety of areas. Inner white matter is composed of tracts of myelinated nerve fibers linking one another, and association fibers, axons connecting nerve cells in different parts of the same cerebral hemispheres, commissural fibers, axons forming between nerve cells of both hemispheres, together forming the corpus callosum. And then we have the projection fibers, axons connecting the cerebral cortex to other parts of the brain and the spinal cord. Optic chiasma. So nerve impulse is carried by cranial nerve number 2, which is the optic nerve, from the right eye, passes to the right side of the brain and to the left side via this organ. This ensures that information from each eye reaches both sides of the brain. So this is where it looks like. So these are the eyes, and then this is the optic chiasma. So if this is the right eye, this is the right visual field. So the information will go to the left side, and on the left visual field, it goes to the right side. And you have here, actually the primary visual cortex is at the back, caudal. Pituitary glands. This is an endocrine gland found below the hypothalamus. And you can find the pituitary gland in the flooring, which is actually near the sphenoid bone. And there's actually a depression on the sphenoid bone called hypophyseal plate, spelled as HYPOPHYSEAL, hypophyseal plate. So that is where the anterior, not just the anterior, including the posterior, the pituitary glands will be located, or is located. Sorry. So we have to go back. Sorry. So that's pituitary. Now we go to your olfactory bulbs. This is the one that I've mentioned. So it's found on the ventral portion of the frontal lobe. So the pair of bulbs which form the most rostral part of the brain, and this is responsible for olfaction or the sense of smell. Infundibulum, the stalk attaching the pituitary gland to the hypothalamus, and the third ventricle invaginates into the infundibulum, forming a recess. Again, hypothalamus. So the hypothalamus is the one that regulates body temperature, and for neonates or newborn animals, this does not regulate their body temperatures as effectively as in adults. So that's the reason why a lot of newborns will die due to hypothermia. Now we go to the midbrain or the mesencephalon. This is located between the diencephalon and the pons and connects the lower brain centers and the spinal cord with the higher brain centers, which is the cerebrum or the telencephalon. So the parts of the mesencephalon, we have the roof or tectum, which has 4 colliculi. So they have rostral colliculi, which is composed of 2 rounded eminences that contain the visual reflex centers. And we also have the caudal colliculi, which 2 rounded eminences behind the rostral colliculi and contain the auditory reflex centers. So the neuronal bodies of cranial nerve number 3 and 4 originate and emerge from this part. Cerebral aqueduct. Ventricular tube running through the midbrain and connects the third and fourth ventricles. And transverse fissure, the deep groove separating the cerebellum from the cerebral hemispheres. Now we are in the hindbrain. So we have the pons. So this is the bulge between the midbrain and the medulla oblongata. Its function includes bridges the spinal cord and the brain and different parts of the nuclei, and the nuclei within the pons working with the nuclei in the medulla oblongata regulate breathing. Cranial nerves 5, 6, 7, and 8 have their cell bodies originate from this location, and cranial nerve number 5 is the only cranial nerve that emerged from the pons. Cerebellum, Latin for little brain. This is the second largest subdivision of the brain, and this is part of the dorsal metencephalon, and it's located dorsal to the pons and medulla and caudal to the occipital lobe, that's lobe, of the cerebrum. Function, motor reflex centers coordinating subconscious and conscious skeletal muscle movement. They also regulate the libido or sex drive of an animal. Parts of the cerebellum, they have vermis, Latin for worm. So this is a central part of the cerebellum. And they have the lateral masses, which are parts on either side of the vermis that are indented to form parallel follici, which increases the surface area similar to the gyri in the cerebrum. Cerebellar cortex, a thin outer layer of gray matter, and they have arbor vitae, which is a white matter tracts branching into the cerebellum. Cerebellar peduncles, these are bundles of fibers connecting the cerebellum to the brain cells. So the caudal cerebellar peduncles connect the medulla oblongata and spinal cord and cerebellum, and medial cerebellar peduncles connect the cerebellum and the pons. Rostral cerebellar peduncles connect the midbrain and the cerebellum. Now we are in the medulla or the medulla oblongata. The caudal portion of the brainstem that connects caudally to the spinal cord, and they have 3 vital reflex centers. They have the cardiac center, which regulates the rate and strength of the heart contraction. Vasomotor center, which regulates the diameter of the blood vessels and blood pressure. So it can regulate if the blood vessel has to constrict or it decreases its diameter, or it can increase its diameter or dilate. So this is where it will happen. And we also have the respiratory center, which regulates breathing. The medulla also serves as the regulating centers of non-vital reflexes such as sneezing, swallowing, and vomiting. They serve also as origin for the cell bodies of cranial nerves number 8, 9, 10, 11, and 12. Pyramids. formed by motor tracts coming from the cerebral cortex on the ventral surface of the medulla, and these tracts cross from one side to the other. Fourth ventricle, this is located between the medulla and the cerebellum. Limbic system or emotional brain, this is from Pasquini and Spurgeon. So this is in the interconnected parts of the cerebellum, hypothalamus, thalamus, and basal ganglia, and other nuclei that deals with emotions such as anger, fear, pleasure, rage, and sorrow. Through these emotions, the limbic system modifies the actions of the animal to help ensure its survival. Reticular formation, again from Pasquini and Spurgeon. A network of nerve fibers and islands of gray matter located in the brainstem from spinal cord to the diencephalon. It is connected to the hypothalamus, basal ganglia, cerebellum, and cerebrum, and it functions in consciousness and arousal. Without the reticular formation, an animal would remain unconscious or unaware, such as comatose. We go to the brain protection. So its physical protection or barrier is the skull, particularly the cranium. It also has the ventricular system. So we have mentioned the third and fourth ventricle, and we also have here the interventricular foramen and these pedicles also. And we also have the cerebellar aqueducts. So the ventricles and the central canal are filled with cerebrospinal fluid or CSF, which is secreted by the choroid plexus. These are a group of capillaries that are found on the roofs of the ventricles. CSF are usually clear, just like plasma in the blood, but they do not have protein. Thus, it is considered as a transcellular fluid. CSF protects the CNS from damage by sudden movement or knock, and CSF also provide nutrients to the nervous tissue of the CNS. We also have the meninges. So 3 membranes surrounding the brain and spinal cord, and the spinal and cranial meninges are continuous at the foramen magnum. So they have 3 layers, starting with the dura mater, also known as the pachymeninx. Pachy meaning thick, or meninx meaning it's a meninge. This is a Greek term. When you say dura, this is already Latin. Outermost of the meninges made of strong connective tissues, and the cranial dura mater has 2 layers. The outer forms the inner periosteum of the cranial bones, thus eliminating the epidural space, and the inner layer extends into the fissures forming the partitions of endural sinuses. And the spinal dura mater has only 1 layer. Arachnoid, so this is the delicate middle meninx. So composed of collagen fibers and larger blood vessels that supply adjacent area of the nerve tissues with nutrients. It has a subarachnoid space, the cavity between the arachnoid and the pia mater where the CSF flows. Then the last one is the pia mater, which is the innermost meninx. We have leptomeninges, which refers to the pia mater and arachnoid together. So when you say lepto, it's Latin for slender. Cisterna magna. So this is a expansion of the subarachnoid space located between the caudal surface of the cerebellum and the dorsal surface of the medulla. And this is where you can get samples of the CSF for clinical testing. So this, sorry if this is quite foreign to you. This is actually in Italian. So when you say pel, that's skin. Perioste, this is the periosteum. Os, that's bone. Dura mater, and then we have the arachnoid mater, and then we have subarachnoid space, and then we have the pia mater. Then we have the blood-brain barrier. So a modification of the neuroglial tissue that supports all the neurons within the nervous tissue. Its function is to protect the brain from substances which are harmful to or not needed by the brain, such as urea, which is a byproduct of protein metabolism, certain proteins, and antibiotics and other drugs. Only oxygen, sodium, and potassium ions and glucose can easily pass through the triple B or the BBB. Now, one of the requirements for a drug, especially our drugs that we use for anesthetics, is it can freely cross the blood-brain barrier. And this is quite selective. This blood-brain barrier is quite selective on what materials are going to get in. However, you really have to consider also that there are certain breeds of dogs, particularly the collie breeds. They have very thin blood-brain barrier due to a mutation. It's a mutation. And that's the reason why certain antiparasitic drugs like the ivermectin and even the chewable form of anti-tick tablets, or the chewable anti-tick tablets, are not allowed to be given to these types of dogs, the collie breeds. There are sometimes it can be presented by some German shepherds. Again, you really have to be very careful. Then we have cerebellar hypoplasia, is a disease seen in kittens whose dams was infected with feline enteritis virus during their gestation. Thus, kittens may never be able to walk in proper coordinated manner. Then we have the hydrocephalus. So there are two types, internal and external. So internal hydrocephalus is due to the accumulation of CSF in the brain ventricles due to the obstruction of the flow of fluid, especially in the cerebral aqueduct from the ventricles through the lateral apertures of the subarachnoid space. And then we have the external hydrocephalus, which is due to accumulation of CSF in the subarachnoid space due to the interference of absorption into the dural venous sinuses. Again, from Skinny and Spurgeon. So this is how it looks like. So this one over here, so this is brain. This is still brain. Now you will see that there's fluid accumulation. So this is an example of an internal hydrocephalus. This is also an example of an internal hydrocephalus, because supposed to be this part over here, only this part over here should be clear or black. This is an MRI of a dog with hydrocephalus. So this is an example of an internal hydrocephalus. And yes, development of, you might be asking, does this impede the development of the dog? Yes, it actually impedes the development of the dog because there's more water, more fluids, fluids rather, instead of water. It's not supposed to be, the fluid should be here only. And this area here should be brain. Also here. This area here should be where the fluids are, and then most of the area will be full of brain activity. But no, this is translucent, meaning it's fluid. So we are done with the brain, and next up you will have the spinal cord. It's quite long, I know. It's quite long. So we're done with the brain. So if you do have questions, please do ask. I really would appreciate it. And till then, see you on the next topic.